Hello, brilliant minds! Ever wondered what tiny molecules carry the grand instruction manual for life itself? Today, we are diving into nucleic acids, the superstar biomolecules that hold the blueprints of living organisms. Quick joke before we begin: why did the DNA break up with RNA? Because RNA was too messenger-y. Now, nucleic acids come in two major types: DNA and RNA. They are essentially long chains, or polymers, made up of tiny building blocks called nucleotides. Each nucleotide is like a three-part toolkit containing a nitrogenous base, a sugar with 5 carbons called a pentose, and a phosphate group that links them all together. DNA's big job is to store hereditary information, the instructions passed from parents to offspring, while RNA is more the multitasker, helping with protein synthesis and turning genes on and off. Nucleic acids aren't just in complex organisms, they're everywhere you look, in all living cells and even viruses. They play starring roles in the amazing processes of replication, transcription, and translation, which are the heartbeats of biological information flow. Imagine the diagram here, a long chain of nucleotides linked like beads on a string, with clear labels showing the nitrogenous base, the sugar in the middle, and phosphate groups connecting the sugars. Welcome back. Let's zoom in on the fundamental building blocks of nucleic acids, nucleotides. Picture a nucleotide as a three-piece puzzle. First, a nitrogenous base that can be either a purine or a pyrimidine. Purines are the taller cousins, with 2 rings, named adenine and guanine, while the pyrimidines are a bit smaller, with 1 ring, including cytosine, thymine, and uracil. Thymine hangs out exclusively in DNA, but RNA swaps it out for uracil. Next up is the pentose sugar, ribose in RNA and deoxyribose in DNA. The main difference? Deoxyribose is missing 1 oxygen atom, giving DNA its deoxy prefix. Last but not least, there's the phosphate group. In a nucleoside, the base and sugar link through a special glycosidic bond. Think of it as a handshake between the base and the sugar. Add a phosphate group to the sugar's 5' hydroxyl group, and voila, you get a nucleotide. Now, imagine these nucleotides hooking together like a chain, forming phosphodiester bonds. The 3' hydroxyl of one sugar connects to the 5' phosphate of the next. This linkage is like the strong glue that holds the chain firmly, giving the nucleic acid polymer its shape and stability. Picture a detailed structural diagram showing atoms clearly highlighted. It helps to see how each part fits and interacts. So, nucleotides are the alphabet of genes, each letter carefully shaped to spell out the story of life. Alright, let's unravel the famous DNA double helix, or as Watson and Crick described it, a right-handed spiral staircase with 2 strands running in opposite directions. The 2 strands are antiparallel, meaning one runs 5' to 3' and the other 3' to 5', held together delicately by hydrogen bonds between bases. Here comes the magic of complementary base pairing. Adenine always pairs with thymine through 2 hydrogen bonds. Guanine pairs with cytosine with 3, making that bond stronger. This pairing is like a perfect lock and key fit that ensures genetic fidelity. The backbone you see twisting around is made of sugar and phosphate units alternately linked, with the bases stacked inside like ladder rungs, adding stability much like how stacking pancakes hold together better than one alone. The helix has a diameter of about 20 angstroms and makes one complete turn roughly every 34 angstroms, dimensions crucial for fitting DNA into cells and allowing it to be copied accurately. Picture a double helix diagram, with base pairs shown as colored steps connecting the 2 sugar-phosphate rails. DNA's elegant structure is nature's way of packaging vast genetic information into a durable yet accessible form. Moving on to RNA, the versatile cousin of DNA. Unlike DNA's double-stranded helix, RNA is usually single-stranded and contains ribose sugar. Plus, RNA swaps out thymine for uracil, a base that fits snugly into its structure, allowing RNA to fold into complex shapes. RNA isn't just one molecule, it has three key types, each with important jobs. First, messenger RNA or mRNA carries copies of genetic instructions from DNA to the ribosomes, where proteins are made. It's the courier of the genetic message. Transfer RNA or tRNA acts like a delivery truck, bringing specific amino acids and matching them up with the genetic code on mRNA via its anticodon region, ensuring the right building blocks come together. Lastly, ribosomal RNA or rRNA forms the core of ribosomes themselves, acting as both scaffold and catalyst to make peptide bonds, joining amino acids into proteins. Now, let's talk about how nucleic acids break down, a process called hydrolysis, which means water splitting. This happens either enzymatically or chemically. Enzymatic hydrolysis uses specialized enzymes called nucleases that neatly snip nucleic acids into smaller fragments, first breaking down the long strands into nucleotides, then nucleotides into nucleosides by removing phosphates, and finally into individual nitrogenous bases and sugars. It's like dismantling a Lego tower block by block. On the flip side, chemical hydrolysis can happen when acids or bases attack nucleic acids. This is less gentle and may break phosphodiester bonds randomly or denature the molecules, similar to how harsh detergents can ruin clothes. In labs, controlled hydrolysis is super useful. Scientists use it for DNA sequencing, forensic analysis to identify suspects, and in genetic engineering to manipulate genes. Time for a quick puzzle. Let's calculate the percentage of each DNA base when given just one piece of information. Suppose a DNA sample contains 30% adenine. What about the other bases? Here's where Chargaff's rules come in handy. Chargaff's rules say adenine equals thymine in amount and guanine equals cytosine. Since all four bases add up to 100%, if adenine is 30%, thymine must also be 30%, giving us a combined 60% for A and T that leaves 40% for guanine and cytosine together. Since these two are equal, each will be 20%. So, the base composition is adenine 30%, thymine 30%, guanine 20%, and cytosine 20%, and they all add neatly to 100%. Let's now zoom out and connect nucleic acids to real-world marvels in genetics and biotechnology. DNA doesn't just pass on traits, it governs how cells specialize and develop. It's the master blueprint for life's complexity. It's also the star molecule in recombinant DNA technology, where genes can be swapped or modified to create medicines or improved crops. RNA species like cRNA and miRNA have taken center stage by regulating gene expression. Think of them as the genome's volume knobs, turning genes up or down. These molecules also serve as therapeutic targets in diseases including cancer. Cutting-edge technologies such as PCR, DNA fingerprinting, and the gene editing tool CRISPR rely heavily on understanding nucleic acid chemistry. They've revolutionized medicine, forensics, and agriculture. Scientists have also crafted synthetic nucleotides and analogs used in diagnostics and to fight viral infections. To wrap up, let's recap the essentials of nucleic acids, the biological information carriers. A nucleotide is composed of a nitrogenous base, a sugar, and a phosphate group, the three components you need to build DNA or RNA polymers. In DNA, adenine pairs exactly with thymine and guanine pairs with cytosine, forming a stable double helix with a diameter of about 20 angstroms and one complete turn every 34 angstroms. These bases connect strands via phosphodiester bonds that link the 3' hydroxyl of one sugar to the 5' phosphate of the next, creating the sturdy backbone. RNA exists in several types, messenger, transfer, and ribosomal RNA, each playing vital roles in protein synthesis. The big picture: nucleic acids are polymers that store and transmit genetic information essential for all forms of life. Their structure and base sequences dictate their function, and the differences between DNA and RNA reflect their unique roles. From genetics to cutting-edge medicine, nucleic acids prove that the smallest molecules can have the biggest impact. That's it for today's journey into nucleic acids. Remember, chemistry is not only about reactions and bonds, it's about the code that makes us who we are. See you next time for more exciting science stories.